The dramatic metabolic improvements observed in rodent time-restricted feeding studies have generated enthusiasm that subsequent human trials have only partially validated, creating a translational gap requiring explanation. Comparative analysis of methodological differences between species illuminates why direct extrapolation from animal models to human recommendations proves problematic and highlights the importance of human-centered research for clinical guideline development. Fundamental circadian biology differences between nocturnal rodents and diurnal humans complicate direct translation. Mice and rats exhibit peak activity and feeding during the dark phase, making restricted feeding during this time aligned with their circadian biology. Many influential rodent studies restrict food access to light phase, creating extreme circadian misalignment, unlike anything typically studied in human time-restricted eating protocols. This misalignment may engage stress response pathways and produce metabolic effects irrelevant to human eating pattern adjustments. Even when rodent feeding aligns with their active phase, the proportion of circadian cycle occupied by eating differs substantially from human patterns, making quantitative comparisons of fasting duration effects problematic. Controlled laboratory environments for rodent research eliminate confounding variables that dominate human studies. Rodents live in constant temperature, consistent light-dark cycles, identical diet composition, and without psychosocial stressors. Baseline diets often involve high-fat formulations promoting obesity and metabolic disease, creating exaggerated pathology against which interventions show dramatic improvement. The ad libitum overfeeding paradigm in which control animals eat continuously and overconsume doesn't reflect human eating patterns, meaning that restriction produces both temporal alignment and calorie reduction simultaneously. Disentangling these factors in rodent models proves difficult, while human isocaloric trials demonstrate that much benefit attributed to timing actually reflects energy deficit. The human environment introduces complexity absent from animal facilities. Sleep quality, stress, social eating occasions, food availability, occupational demands, and circadian preference create heterogeneity in responses and adherence that animal models cannot replicate. Individual variation in chronotype, baseline metabolic health, microbiome composition, genetic background, and lifestyle factors produces the broad response distribution seen in human trials, contrasting with relatively homogeneous inbred rodent strains. These sources of variation explain why human trials show modest, variable effects while rodent studies demonstrate robust, reproducible outcomes. The implications for research translation emphasize that animal models serve to generate hypotheses and explore mechanisms rather than predict clinical effect sizes. Human trials with appropriate controls and attention to real-world context remain essential for clinical guideline development. Researchers should resist overinterpreting animal model findings when proposing human interventions and acknowledge limitations when extrapolating between species. The media and public should understand that studies in mice show represents early-stage exploratory research requiring human validation before informing personal health decisions. The translational gap between rodent time-restricted feeding and human time-restricted eating serves as a valuable case study in the challenges of translating animal research, reinforcing the necessity of species-appropriate evidence hierarchies in evidence-based medicine.